This PowerPoint presentation is looking at a specific portion of the cardiovascular system, the blood. This is going to be part one of two in terms of video recordings. You'll recall that the cardiovascular system includes the heart, blood, and blood vessels. A single drop of blood contains millions of red blood cells, white blood cells, and platelets. There are three main functions of blood, including transportation, defense, and homeostasis. In terms of transportation, remember that whenever we eat food, those nutrients that come from the digestive tract go into the bloodstream and will go to the liver for further processing and then be sent throughout the body to deliver those nutrients to other cells. Oxygen coming from the lungs to the heart will also go through systemic circulation to deliver oxygen to cells throughout the body. And not only do we have nutrients coming in, but cellular waste and byproducts has to circulate through the body in order to be removed by the lungs when we breathe out carbon dioxide or through the urinary system. Hormones have to circulate through blood in order to reach their target cells. When it comes to defense mechanisms, there are formed elements of blood known as white blood cells or leukocytes. These white blood cells are important because they help destroy bacteria, help us fight against viruses, and also help destroy cancerous or mutated cells. There are also broken down pieces of cells known as thrombocytes, which form platelets. And in combination with platelets, plasma proteins can defend and protect us against the loss of blood. Homeostasis comes in three different ways. Our blood can help us regulate our body temperature, and that's because there's a large water component in blood, and water is a good medium for heat. Our blood also helps us regulate our level of acidity, our pH, and blood also helps us regulate how much water is in our bodies. Remember, blood circulates through kidneys 24 hours a day, 7 days a week, and so if we have too much water in our bodies, then that excess water would be excreted. If there's not enough, then the kidneys, in concert with our hypothalamus, can help hold on to water. When we look at the composition of blood, remember that blood is a special type of connective tissue, and all connective tissue has cells and an extracellular matrix, which is the stuff surrounding the cells. In blood, we refer to cells as formed elements, and those formed elements include red blood cells, known as erythrocytes, white blood cells, known as leukocytes, and platelets, which are broken down pieces of thrombocytes. The body has to make cells. That's why they're called formed elements. The body formed them. And then the extracellular matrix, the stuff surrounding the cells in blood, is referred to as plasma. Remember, if we were to take a blood sample and spin it around real fast in a machine called a centrifuge, then the blood would actually separate. Normally, blood makes up about 8% of your entire body weight. When we centrifuge a sample of blood, we see that there's three separate and distinct layers. There's a layer at the bottom that looks red because that's where the red blood cells are found. That's known as the hematocrit. Just above that layer of red blood cells or hematocrit, we see a special layer of white blood cells and platelets called the buffy coat, and above that is where we find blood plasma. And about 55% of a blood sample is usually blood plasma, give or take a little bit. About 45% is the hematocrit or red blood cells, And then the buffy coat, which is the platelets and white blood cells, is usually less than 1%. If we look more closely at the blood plasma, we see that it's mostly water. 91% of that is mostly water. There are also plasma proteins, and plasma proteins include things like albumins, globulins, and fibrinogen. Albumins make up the bulk of plasma proteins, and these plasma proteins are really important because they help maintain fluid in the bloodstream when it gets filtered through the kidneys so that not all the water gets peed out. And when we look at the plasma, there are other solutes in this fluid, including electrolytes like sodium and chloride and magnesium and potassium, and nutrients as well. And we also have waste products like urea in the plasma portion, dissolved gases like nitrogen, and other regulatory substances like hormones. When we look specifically at the white blood cells, there are different types of white blood cells, and we're going to learn about those white blood cells. Those include neutrophils, lymphocytes, monocytes, eosinophils, and basophils in the order of most abundant to least abundant. Again, that area in the centrifuged sample of blood that is the deepest in the test tube and has that reddish appearance is known as the hematocrit, or packed cell volume. In normal blood, that's about 45% of the entire sample, and it's basically a sample of red blood cells or erythrocytes. Now, the 45% is a range. Men tend to have a higher level of hematocrit, and it goes anywhere between 42 to 52%. Women have a lower volume of hematocrit, which is normally 37 to 47%. Again, if you remember that the normal range for hematocrit or packed cell volume is 45%, that's good. The plasma percentage refers to the rest of the blood sample that's not the hematocrit. In this particular picture, you can see that in anemia, we have a lower hematocrit percentage. In polycythemia, we have a higher hematocrit percentage. We're going to take a look at those blood disorders a little bit later. This takes us to more characteristics of blood. So when we look at blood, blood normally has a bright red color when the hemoglobin is oxygenated. Now, in arterial blood, there are higher levels of oxygen than in venous blood because arterial blood is that freshly oxygenated blood that hasn't delivered oxygen to the rest of the body's cells. However, venous blood is that return in the system that's coming back to the heart so it can go back to the lungs to get more oxygen. So venous blood is not as bright red as arterial blood. And whenever oxygen binds to that iron portion of hemoglobin, that's what turns it that brighter red color. Blood is thicker than water. It's about 5 times more viscous than water, and it has a slightly higher temperature than your body temperature. The pH of venous blood tends to be lower because it's slightly more acidic because of the carbon dioxide than arterial blood. Arterial blood tends to be a little bit higher, so it's a little bit more basic than venous blood. If you were to take an average between the two, the pH of blood tends to be about 7.4. Again, the volume of blood that makes up the percentage of your total body weight is about 8%, which is equivalent to anywhere between 4 to 6 liters of blood in your body. Remember that plasma isn't just water. About 91% of plasma is water. However, we have plasma proteins, which are proteins found in that liquid portion of blood, and they make up about 7% of the plasma. There are 3 different types of plasma proteins, including albumin, which is the most abundant of all the plasma proteins. It helps transport lipids throughout the blood and also is a big contributor for osmotic pressure, so that when blood flows through kidneys, not all the fluid goes into the tubules of the kidneys to potentially be flushed down, because the osmotic pressure of the albumin helps pull water and keep it into the bloodstream so we don't lose all our fluid and urine. Globulins come in 3 flavors. We have alpha globulins, beta globulins, and gamma globulins. The alpha and beta globulins help transport things like iron, lipids, fat, soluble vitamins, which include A, D, E, and K, as well as contribute to osmotic pressure so that not all the fluid goes through the kidneys into the potential toilet. And then gamma globulins are important in our immune system. And lastly, fibrinogen is a special plasma protein that helps become soluble when a blood vessel breaks to help in the process of clotting. Fibrinogen will turn into a fibrous protein known as fibrin, and we'll see that process soon. As you look at this slide, you can see a picture of a centrifuge test tube to the upper right-hand side, and then the chart on the left demonstrates the components and percentages of blood. So remember that hematocrit is normally 45% plus or minus, and then the plasma would be 55% plus or minus. Remember that the white buffy coat of a centrifuge sample consists of white blood cells and platelets, and that the biggest component of plasma is water, but we do have plasma proteins, albumin being the most abundant, and then globulin and fibrinogen. And you should know about the functions of those plasma proteins. Remember that formed elements include things like red blood cells, white blood cells, and platelets, and those are produced by the bone marrow and sometimes further matured or produced in lymphatic tissue as well. So this is a nice chart. It shows you where things are made and what their functions are and what the percentages of things are in blood that you'll find. The term hematopoiesis means formation of blood, specifically blood cells, formed elements. The term poiesis means formation of. Hema, blood. So when we look at how red blood cells, white blood cells, and platelets are formed, when an egg and a sperm come together and conception occurs, initially the yolk sac, which is a special structure that helps deliver nutrients to that growing embryo and fetus, is going to be the primary site of development of red blood cells, white blood cells, and platelets. And as that embryo turns into a fetus, the liver and spleen join in, as well as lymphatic tissue. So we've got all of those sites contributing to hematopoiesis. And then as that individual grows into a child, the job of hematopoiesis is taken over by the long bones, where there's a lot of red bone marrow. And then as the individual ages, remember that the ratio of fat to red marrow in long bones changes, and in adults there's more yellow marrow than red marrow in terms of the ratio found within the long bones. We still do have the epiphyses of the femur and the humerus that contribute to hematopoiesis, as well as the bodies of the vertebrae and the flat portions of the cranium, the pelvis, and the sternum that contribute to hematopoiesis. And in the adult, what's kind of interesting is that if a person were to start hemorrhaging, then some of the yellow marrow, the fat in the long bones, can actually reconvert into red marrow to help create more red blood cells. And it takes a while, but that can happen in the event of severe blood loss. Okay, so why do we need to continuously produce these formed elements? Well, they don't live very long. They have a very short lifespan. In fact, red blood cells only live for 120 days. So we continuously need to produce these formed elements, with the exception of memory cells. They're special white blood cells that we'll discuss later, and they live longer, but all formed elements, with the exception of those memory cells, have to be replaced continuously. Now, in the event that there is a person who's dealing with any kind of cardiovascular disease, then in the adult, the liver, the spleen, and the thymus may contribute to the production of formed elements, or hematopoiesis, and they call that, or refer to that, as extramedullary, because it's not happening in bone marrow when the liver, the spleen, and the thymus have to kick in to help out. This picture is really helpful in understanding how cells are formed, because they have to come from stem cells. And if we look at this picture on the lower left-hand side, you'll see that a piece of bone with the medullary cavity has been enlarged, demonstrating hematopoietic supportive cells. Those are the green ones. They're called stroma. Those are the supporting cells. But then we also see there's an adipocyte, that's a fat cell, and a blood vessel, and then you can see an arrow going up to the right of the box where it points to a cell that's called an HSC, a hematopoietic stem cell. And when that cell divides and creates more of itself, one of the two cells that is formed during mitosis is going to turn into what's called a multipotent or multipotential stem cell. And this multipotential or multipotent stem cell can turn into a lymphoid progenitor cell that can turn into either natural killer cells, T lymphocytes, or B lymphocytes. Another direction that this multipotential stem cell might take would be that of a myeloid progenitor cell, where this cell can be turned into neutrophils, basophils, eosinophils, monocytes or macrophages, platelets, or red blood cells. As you look at this picture, you'll also notice that some of the stromal stem cells, before they divide into hematopoietic supportive stroma, can also turn into bone or cartilage. And by the way, the term cartilage is misspelled in this picture, unfortunately. But they can turn into pre-osteoblasts, which will turn into osteoblasts. There's a possibility that they may turn into skeletal muscle cells or hepatocyte stem cells. They're still researching that, but that's a possibility. So in the process of these cells differentiating, remember, we start with a stem cell. It divides and becomes a little bit more differentiated in terms of it's going in a certain direction. So the multipotent stem cell can either become a lymphoid progenitor cell or a myeloid progenitor cell, and those progenitor cells are going to further differentiate into the final cells that they will become. So when we look at what determines the direction that those progenitor cells take, it's really determined by different and specialized chemicals known as hematopoietic growth factors. Erythropoietin is a hormone protein that is secreted by the kidneys when there's not enough oxygen in the body. That's a condition known as hypoxia, and that helps direct the production of more red blood cells so that more oxygen can be carried. Thrombopoietin is a special growth factor that helps direct those progenitor cells to turn megakaryocytes into platelets, and that specific growth factor is secreted by the kidneys and the liver. There are also special chemical messengers known as cytokines that are either autocrine or paracrine signalers that cause the proliferation or creation of more progenitor cells. Proliferation is creation of a lot of things. So bunny rabbits are highly proliferative. They create more of themselves. When we look at colony stimulating factors, those are growth factors, specialized growth factors that trigger the differentiation for many white blood cells and platelets to occur. And then there are also chemicals known as interleukins, which communicate among white blood cells and help to mediate inflammation. Because remember, white blood cells are going to be released when there is some kind of pathogenic invader or damage. Now, erythrocytes, which is the fancy name for red blood cells, erythrocytes account for 25% of all the cells in the human body. And they not only transport oxygen, they also transport carbon dioxide. They don't leave the blood vessels, and when they are mature, they take on an internal concavity on both sides. That's why it's referred to as concave, biconcave discs, and that allows for a maximum surface to volume ratio. Why would that be important? Well, the more surface area there is, the easier it is for oxygen to diffuse and carbon dioxide diffuse from the outside of the red blood cell to the inside where it can bind to a carrier. They're also very flexible. Well, why do you think that is? Well, these blood cells have to be able to bend and move through tiny, tiny little spaces. Remember that capillaries are the tiniest blood vessels in our body, and you can fit 10 capillaries in one strand of hair. So sometimes the way that capillaries twist and turn and bend, these red blood cells have to be flexible to move with the shape of capillaries. They are also anucleate, which means there's no nucleus in them. It actually gets extruded shortly after they leave the bone marrow. And they don't have organelles except for a few protein fibers that help make up the cytoskeleton for them to maintain some little bit of shape. And again, why would that be? Well, that makes more room for hemoglobin to transport oxygen and also carbon dioxide. So when we look at hemoglobin, and this is a picture of a molecule of hemoglobin, we see that one molecule of hemoglobin contains 4 globin proteins. Those are the different colors. So we've got bright red, light brown, darker brown, and kind of pink. Those each represent a globin protein, and each globin protein is bound to a molecule of an iron-containing pigment known as heme. And in the heme disc, you can see that iron in the very center. And a single red blood cell can contain up to 300 million hemoglobin molecules and carry more than 1 billion oxygen molecules. When it comes to oxygenation on hemoglobin, whenever we see that bright red color in blood, that's what's referred to as oxyhemoglobin, because when oxygen binds to the iron portion of the heme group, it turns it that bright red color, and that's what we call oxyhemoglobin. Deoxyhemoglobin is going to be a darker red color. That's what we find in venous blood because it has released its oxygen, and then when carbon dioxide binds to amino acids in hemoglobin, it's referred to as carbaminohemoglobin. And remember we said that carbon dioxide can bind to hemoglobin, and Hemoglobin is a protein made up of amino acids, so the carbon dioxide actually binds to amino acids in hemoglobin. Hemoglobin imbalance can occur if there's either not enough hemoglobin or too much hemoglobin. Anemia is what occurs when there are either insufficient numbers of erythrocytes or insufficient hemoglobin, and the result of that is that the tissues throughout the body become deprived of oxygen. On the opposite end of the spectrum, polycythemia occurs when there's an overproduction of erythrocytes in the body, and that makes the blood thicker, so it increases blood viscosity. What you see on this particular slide is a picture of a person's finger in a pulse oximeter, which is a small machine that shines light through the finger, and it measures the percentage of hemoglobin sites that are occupied by the oxygen in a person's blood. And that value is what we refer to as a percent saturation or percent sat. Sometimes it's also referred to as O2 sat, or sometimes even a pulse ox reading. And because the hemoglobin absorbs light differently when it's oxygenated, that machine can detect the difference and determine how much oxygen is present based on how much light is able to pass through the tissue. The term hypoxemia refers specifically to low levels of oxygen in the blood. Hypoxia is a generic term for low oxygen levels. And knowing that the kidneys filter our blood 24 hours a day, 7 days a week, which ends up being about 180 liters of blood every day, those are really good organs for monitoring the percent saturation of oxygen in the bloodstream. And when oxygen levels are decreased, there are special cells in the kidney, they're called peritubular cells, and they're most likely going to be found in the endothelium of the cortex. Remember, in an organ, the cortex is the outer portion, and then the medullary portion is the inner portion. So it's thought that these endothelial cells of the cortex and the outer part of the medulla of the kidney produce most of the hormone erythropoietin. And once those oxygen levels come back up to normal as a result of erythropoietin stimulating the production of more red blood cells in the bone marrow, then that turns off the system. Again, that's another example of negative feedback. So let's just take a look at the picture here on this slide. You can see that at the top left, it says the peritubular cells in the kidney detect a decrease in blood oxygen levels. So they secrete erythropoietin, which is a hormone, into the blood, and as it circulates through the bloodstream, when it reaches the bones, and remember that we have a lot of marrow as adults in the proximal epiphyses of the humerus and the femur, that erythropoietin hormone will stimulate the proliferation, so we're going to make a lot more of, and differentiate the erythroid progenitor cells into more committed cells called reticulocytes, and it prevents cell death. Remember that cells have a normal lifespan, and so the erythropoietin actually helps prevent the apoptosis where the cell would normally die. So now we have more reticulocytes entering the circulation of the blood, and they start to differentiate into erythrocytes by getting rid of the nucleus, and the organelles inside those cells also starting to basically dissolve, and also those pieces parts at that point would be extruded, and then there's more erythrocytes at that point that are able to carry oxygen throughout the body to the different tissues in the body. Now, remember that not just the nucleus is kicked out of that mature red blood cell, but there are no more organelles. It's just a little bit of cytoskeleton to help maintain a little bit of the shape of the erythrocyte. And that's a good thing. If you remember that, if we had mitochondria in there, they would be using the oxygen. And so it's important not to have mitochondria in a mature red blood cell. So if someone were living in the mountains versus down by the seashore, what would be the difference in that person's hematocrit level? Hopefully you'd expect the hematocrit level to be higher in a higher altitude, because remember that the pressure as we go up in altitude decreases, and so we need more red blood cells to circulate the amount of oxygen that's found in higher elevations. When it comes to erythrocytes, there are certain things that the body needs to be able to put these cells together. For instance, we need iron for the heme groups. We need copper because that helps the body make heme. And we also require zinc because zinc acts as a coenzyme to, again, produce the heme portion of hemoglobin. And then we also need B vitamins, specifically folate and B12, because those help in the process of DNA synthesis, which occurs during mitosis. And if we're having mitosis occur with hematopoietic stem cells dividing and dividing, then obviously we need the B vitamins as well. When we look at the life cycle of an erythrocyte, understand that our body produces more than 2 million of these cells every second. So obviously we're going to need all the pieces parts to be able to put these cells together. And after circulating for 120 days, give or take a few, those erythrocytes become old and damaged and worn out because they have to squeak through little places. And so that's why we need to replace those cells, and also we are able to recycle the pieces parts to rebuild and make new ones. If we take a look at this picture on this slide, you can see the beginning of the life cycle of an erythrocyte in the upper right hand corner, where it shows that hemopoiesis or hematopoiesis begins in the bone marrow, where we have hematopoietic stem cells that will undergo mitosis and divide, and some of those cells, half of them, will remain as stem cells and will continue to divide, and the other portion will go on to become differentiated into erythroblasts. As erythroblasts undergo mitosis, some of them will remain as erythroblasts and some will further differentiate into immature red blood cells known as reticulocytes. And it's actually the reticulocytes that will go into the bloodstream, and when they enter the bloodstream, they actually extrude or get rid of their nucleus so that there's more room for hemoglobin, and so that the cell can also flatten out, become that biconcave disc that needs to circulate throughout the bloodstream. These erythrocytes, as they go through circulation and as they become older and damaged and worn out, may become phagocytized or just break apart, but oftentimes they're phagocytized by macrophages in the liver, the spleen, and in the bone marrow, where the macrophages, when they engulf the red blood cells, will further break them down with the digestive enzymes from their lysosomes. And when that occurs, the globin, the protein portion of the hemoglobin, will be broken down into amino acids, and those amino acids can be sent back to the bone marrow to be used in the production of new erythrocytes. And any hemoglobin that is not phagocytized, because sometimes these erythrocytes can just break down in the bloodstream, so any of the hemoglobin that's not phagocytized will be basically broken down and will release alpha and beta amino acid chains that will be removed from circulation by the kidneys. Now, the iron contained in the heme portion of hemoglobin will either be stored in the liver or spleen, or it will be carried through the bloodstream back to the red bone marrow to be recycled into new erythrocytes. The non-iron portion of heme is further broken down, and it's broken down into a waste product, a green pigment protein known as biliverdin, and the biliverdin can also be converted into a different protein pigment known as bilirubin, which is a yellowish orangey protein pigment. And bilirubin will bind to albumin in the bloodstream, and it will travel in the blood to the liver, which will use that to create bile. And bile will eventually be released into the digestive tract from the gallbladder. And when it's in the intestines, the large intestines have bacteria which will break the bilirubin apart from the bile, and they'll convert it into urobilinogen, which will then be converted into stercobilin, and then it's eliminated from the body in fecal matter. And that's how fecal matter gets its brown color, from the stercobilin. And the kidneys will also remove any circulating bilirubin and other related metabolic waste products like urobilins, and it will secrete them into the urine, and that's what gives urine its yellowish color. So again, the broken down pigments from the hemoglobin being destroyed can show up as a greenish pigment. If there's bruising under the skin, it might be greenish or purplish, and that's from the biliverdin. We know that the bilirubin, the yellow pigment protein, will bind to albumin and break down into urobilinogen and stercobilin. The stercobilin gives the brown fecal color to feces, and the urobilin provides that yellow color in urine. And what you see at the very top left corner of this series of pictures is a condition of an overaccumulation of bilirubin, because normally, again, the liver would help process and create bile with the bilirubin. If the liver's not functioning properly, then there can be an overaccumulation of bilirubin that starts circulating throughout the body, creating a yellowish condition of the skin, the mucous membranes, and can even be seen in the whites of the eyes, and that's known as jaundice. So if you ever have a patient who presents with yellow skin, yellowish mucous membranes, yellowing in the white of the eyes, that could potentially indicate there is a problem with the liver. This ends the first part of blood.